speaker is Yoshiyasu Fukuyama from Tokushima, Bunri University, Japan, and his topic is neurotrophic compounds of Javanese, Zingiber, purpureum. And I will introduce this professor. Fukuyama has completed his PhD degree in chemistry from Osaka City University and then spent 3 years as a postdoctoral fellow at Oregon State University. He moved to the Institute of Natural Products Chemistry at the Osaka Pharmaceutical Company. He has been working as a professor at Tokushima, Bunri University since 1988, and now is Dean of the Faculty of Pharmaceutical Sciences. He has published more than 220 papers. Wow. In reputed journals and serving as an editorial board member of CPB, quite famous journal in this area, right? His current research interests focus on chemistry and biology of neurotrophic natural products. He is a recipient of Tokushima New Paper Award in 2006. Welcome.
Thank you, introduction, Professor Chan. I feel a little bit tired because I'm doing this chair, this session from morning. I'm tired. I'm sorry. Anyway, let's try my best to talk about my presentation. First of all, I would like to express my sincere thanks to Overnight Committee of Pharmacognosy 2014 and Professor Asakawa for giving me. Professor Asakawa told me that why don't you attend this meeting with Professor Asakawa? Okay, I agreed because I have so many friends in Beijing, so I'm really happy to see them again. So today I would like to introduce you interesting edible Indonesia ginger, which has a very interesting neurotrophic ingredient. And also I like to talk about not only what active ingredient are and not also but also about neurotrophic efficacy of these active compounds. The neurodegenerative disease, namely we say dementia, that caused by several causes, such as brain injury and vascular disease and Parkinson's disease and so on. But about 56% of dementia are due to Alzheimer's disease. At the present time, only 4 drugs shown here are used for the treatment of Alzheimer's disease. This is the first drug, donepezil. Last year, these 3 drugs, galantamine, inversigimine, and memantine, were launched into a market. These drugs, choline acetyl, most of them are choline acetyl inhibitor. That means they can't prevent neuron from progressing by neurodegenerative disease and cannot stop the disease of neurons. They have a big problem. This is showing neurotrophic, what we call neurotrophic factor, such as NGF and BDNF, etc. they are well known to play a very important role in differentiating stem cell of a neuron to neuron, and the enhancement neurite autogrowth, and also maintain proliferation. The very, very important proteins to contribute the leaf of neuron in our futures, in the life. But neurotrophin has a very serious problem for treatment of Alzheimer's disease because they cannot pass through the blood-brain barrier and not stable under physiological condition because of high molecular proteins. So I have my laboratory that decided to discovery to find out small molecule which has a neurotrophic property like neurotrophic factors. For finding out neurotrophic mimic small molecule compound, two cell system are used. Now, one is the PC12 cells. Maybe you know very well these cells. The PC12 cells is usually proliferating. It's kind of a tumor cell. And in the presence of a neurotrophic factor, stop neurolifration and differentiation into neuron-like cell. This one does, which has many neurite rings here. So we call this kind of activity neurogenesis. The other screening cell is rat, primary culture, rat cortical neuron. This is a real neuron. If compound shows a neurotrophic factor, this cultures the neuron enhance it into have a long neuron and a neurite. So we can see morphologically how active our compound has. So we are using both systems. We have continued to find out, search for neurotrophic compound of a natural product. I'll show you our several neurotrophic compounds which we have found so far. So these are typical compound. Maybe you know some compound. For example, the merlactin A and the gyrenin, neobimacerin. It's very structured, very interesting, and also activity is very, very fascinating. So many organic chemists in the world try to synthesize this compound. So today, I've just introduced special ginger we call it Bangle. It's a botanical name in the Zingiber purpureum. This is an edible plant, and it is used as a spices in Asia, and also used for traditional Indonesian medicine, Jamu. Maybe Indonesian people know very well about Jamu. And Jamu is very, very popular in Indonesia to get energy, and also used for fever and headache reliever and so on. And especially they use it for woman after childbirth. Anyway, if you take a Jamu, you're supposed to get energy to become powerful, I hope. We found the matrix of Bangle showed neurotrophic activity. This is a PC12. without anything, NGL and any compound. That means this kind of cells. But if add a metal extract of 25 milligram per liter in this PC12 cultures, you can see that these are new right growth sprouting. That means that something induce neuritogenesis. So we have decided to identify what they are in the Bangle by activity-guided fractionation. After all, we could isolate, this is a fractionation. So the activity showed the fraction of 5, 6, 7, 8 showed good activity. So we try to isolate the compound from these active fractions. After all, we could isolate so many compounds, about 26. It's a very interesting compound too. This is a new compound, coumarin the diamond. And among them, only neurogenesis activity of compound 1 and compound 2. The other compound did not show any neurotrophic activity without energy. But several compounds showed neurite auto growth promotion in presence of energy. So these compounds only like NGF-like compound. I'll show you the how good these compound is against the PC12. This is showing the PC12. This is the control. This is the presence of NGF. This is the presence of compound 1 and compound 2. The concentration of the 30 micromole. You can see NGF and these compounds enhance the neurite growth sprouting here. This is nothing. Maybe easy to find out how good it is. This is NGF. This is a vertical line that's showing the percent of cell with neurite number. So mean compound 1, the compound 1, 2 enhance neurite numbering, neuritogenesis here. It seems to me that compound 1 transform a little bit more potent than the cis one. This is just to measure the length of a neurite. That one is the number of neurons from cell body. See, this is NGF. This is the length of neuron, it's vertical line. And this compound can enhance neurite length at a concentration ranging from 0.05 micromolar to 30. Pretty good. But the PC12 cell is a cancer cell, but not real cell. This cell is very good, responsive to neurotrophic factor like NGF. So this is a very good cell line to pick up the active compound of a neurotrophic tree. But I like to use second screening system, which is a real neuron. This is a protocol for screening. We take a neuron from a baby, the brain, the primary culture shown here. This is a result of primary culture of neurons. This is a control. It's not easy to see clearly. Very small length of a neuron outright. And this is a BFGF as a control. And this is a compound 1 and compound 2 at some 3 micromolar. It seems to be the neurite length is enhanced pretty good, significantly. So quantitative analysis of neurite growth, compound 1 to analyze it. This is the control. This is a BGL. BGL is pretty good because this is a growth factor. Enhance very high the length of neuron. But this is compound, this is compound 2. from the concentration 0.03 to 3 enhance the length of neuron. But seems to be not in load dependent. I don't know why in this case. Anyway, enhance the neurite length by our both this compound. This is showing the number of sprouting cell body. This is very important because this compound causes neuritogenesis. This is BHL, see compound 1, 3, like a BGL, enhance neuritogenesis, pretty good. So I know this compound is a pretty good neurotrophic candidate by estimating PC12 and cell line in vitro. I really want to confirm in vivo the experiment, how it is okay or not. So we try to synthesize this compound. You can see very easy, this butadiene and dimerized by this delucidated reaction. We try to find out several conditions. After several trials, we could find that this entry 5, because that is a compound 5 DNA is refluxing in paraxylene for 12 hours to give us 86%. Very good, very simple. So now we have a large amount of this compound in hand. So we are ready to carry out in vivo experiment. So to this end, we have decided to use OMICS mouse, olfactory valve ectomized mice. This is a mouse brain. This part of olfactory valve, which very important, responsible for sense of a neuron. So if remove this one, we can't detect it any kind of sense, neuron. So we remove this one. We say OB marks. This OB marks is thought to be acute to become memory impairment depression. That means it's regarded as a kind of a neurodegenerative model mouse. So we apply this OBX mouse, compound 1 and compound 2, and the fluoxetine as a positive antidepressant drug. And the schedule right here, OBX mouse at day 40, compound 1, 2, 50 milligram per kilo per dose, every day through to 20 days, PO, and then 21 BRDNL injected, and then 28 the animal OBX as a sacrifice. This is the brain. This part cat. This part is the DG, dental gyrus region of the hippocampus. So this hippocampus region stained immunohistochemical chemistry, and then we analyze it confocal laser scanning microscope. So these 3 pictures, let me explain what are they. These blue one indicated newly generated cell, including all kinds of cells, of course, including neuron cell. This red one, neuron, all of a neuron. And then both are merged. This is a merged picture. These are pink, red, yellow colored part indicating a newly generated neuron. I mean, the neuron is generated. It is fantastic. So this is the result. This is the OBX mouse. Only 0.5 CMC is administrated here. So please compare this one with this one, this one, this one. This is a fluor active compound. This is our compound 2. You can see these are yellow spot. These are yellow spot, or nothing. The yellow spot, number of yellow spot means increasing the neurons are regenerated. This is a quantitative analysis. We counted the degenerated neurons. This is no mean the non-operated mouse. Sham mean the operated mouse with OBX. This is the OBX. No, OBX left. This is OBX. This is compound 1, compound 2. And compare with the B group, you can see compound 1 induce newly generated neuron. So this is a neurogenesis. So it's a very interesting result for me. So I think that's all I want to talk about you. So our compound, we found the neurotic compound in Bangle. That is both this kind of simple compound. This compound could induce new differentiation from stem cell to neuron. I mean, regeneration, and also enhance neuritogenesis growth and maintain neuroprotective activity. That means Bangle, Indonesia, ginger is very expected to develop as a food or food additive for protecting neurodegenerative disease. Nothing. Thank you very much.
Very good research. Any question?
Did you take this expression of neurospecific marker?
In what? Specific marker?
For example, so neurotransmitter receptor expression.
Actually, mechanism action, the study now going on. I'm not sure to answer your question. As far as I know, the recent result, this compound probably targeting at the track phosphorylation inhibited. You may understand? Usually, neurotropic factor has a receptor, track A and B, the B2. And the NGA track A, and the BDNF track B. Then this compound closely relates to track B, BDNF, but no binding at the receptor at all. But phosphorylation of the track is very important to keep showing some activity. But the phosphorylation immediately dephosphorylated by enzyme. Probably this compound may stop, protect the phosphorylation. This is only I know.
So you mean so you check the indirectly? Okay.
And your last question.
In your OBX model, you tested only one dose, right? How did you know that? In your OBX model,
Yes.
you tested only one dose.
Yeah, one dose.
How did you know that that was the correct dose to test?
Yes, good question. Yeah, we need to do the several doses. Yes, it is. So how we are doing that? Because
you are lucky.
Yeah, lucky. Yeah, several times try. Yes, we need those dependent matter. So far, no. So yeah, we have to do that.
Sir, just a minute, sir. A simple question. There is any basic similarity between the drugs that is already established in the market for such similarity to your compound, isolated compound.
You mean the already
Structural similarity between the already established in the market drugs?
So for example, there is some is a drug, you mean?
No, I don't know. I don't know what is the drug actually. But is there any similarity, structural similarity that may base of your study?
Yes, we should do. Because now, this point, I'm really interested in the whole extract of a Bangle. Because this is a food, you know. But first, for development of as a functional foods, we have to identify what active principles is. This is a result, you know. But if try to develop active plants into drug, it's a very long way to do that. So first I am thinking the Bangle leading to the function drug. So I don't worry about the other compound. Only I'm interested only this active principle. But if I want to develop this compound into drug, I have to do your research. Yes. Okay.
Did you mention that the active site of the two compounds?
Yes, I answered to him.
Also, okay.
Thank you again, Fukuyama. And